The special middle seats are for the latecomers. My name is Kristen, and I'm one of the organizers for the Seminars Quantum. I co-organized with Lychee and also Terrence Wang here today. And we are representing the Quantum Lab. We'll start with our land acknowledgement. The University of Washington acknowledges the Coast Salish peoples of this land, the land which touches the shared waters of all bands and tribes in the Suquamish, Tulalip, and Muckleshoot nations. Our lab also acknowledges that we live and work on the ancestral lands of the Duwamish people, first people of Suquamish. No complaints online, so I'm just gonna keep going. Today we are going to hear from Juliette Champagnat, our new postdoc, who just only started on March 1st, so it's 1 month in, and she is here in the Pond Lab. She's collaborating with the Alaska Fisheries Science Center to develop state-based stock assessment models for the Gulf of Alaska stocks, and she's interested in identifying environmental drivers of productivity for stocks. She earned her PhD in Brittany, France, and she will present some of her PhD results today, so not all the state space environmental stuff. You're going to have to give her longer than a month to hear about that, but we will hear about what she did for her PhD. Juliette will take questions at the end of her talk, so we got to start wrapping them up. Folks online, you can submit questions throughout the talk into the Q&A box, but I will not read them until the end. And with that, thank you for coming, and Juliette, thank you for volunteering to share your work so early. Help me welcome Juliette. Might have to re-click on the path.
Yeah, I think it's okay. Okay, thank you all for being here. Thank you, Christine, for the introduction. So yes, today I'm going to share a bit of my PhD results, which was about modeling the impact of the anthropogenic pressure exerted on the essential fish habitat for the population dynamics of marine exploited species. I did my PhD in Rennes, in Brittany, in France. We're under the supervision of Olivier Lepape and Etienne Rivaud, who some of you know. And I was funded by the French Office for Biodiversity and the Seawise European Project for a part of my PhD. So let's start with a definition of the essential fish habitat. So there is a big, long definition in the Magnuson Act that can be summarized by that habitat which support at least one stage of life. And if we look at this life cycle of marine species, you can identify 5 essential habitat: adult feeding area, spawning grounds, larval drift, juvenile habitat, and the migratory pathway between them. And so that's a lot of habitat. So for prioritization purpose, you might want to identify some critical habitat. And if you use some criteria of spatial restriction of habitat and concentration of individuals within this spatially restricted habitat, you can identify spawning ground and juvenile habitats that as essential, critical essential fishing habitats. Migratory pathway might fall within this definition as well, but I didn't study that in my PhD. So those EFH are subjected to a lot of anthropogenic pressure. They are threatened by anthropogenic and natural pressure. And this is particularly true for the coastal areas where there is a lot of spawning ground and juvenile habitats. And there is a lot of species using coastal habitats. So they represent 44% of the species assessed by ICES, the European Council for the Exploration of the Sea. And so this is also 3/3 of the commercial landing in the Northeast Atlantic. I don't have the figure for North America, but I guess it's kind of the same ratio, right? And 92% of the species are impacted at one stage of life and 38 in multiple stage of life. And so here we have a little diagram on the importance of each category of human pressure. So you have toxicant pollutant, hypoxia, invasive species, coastal development, and indirect fishing impact. So there is a wide range of anthropogenic pressure affecting fish in the AFH, and this is widely acknowledged. There is a whole bunch of studies, for example, studying the impact of pressure on juvenile and impacting condition, growth, and survival of the juvenile, but which is a bit still missing at the consequences at the population scale. So trying to get from individual to population scale, and this is still poorly quantified. And it would be very useful to guide effective management actions. So that was the aim of my PhD, modeling the impact of anthropogenic pressure on the FH for the population dynamics. And so I will show you a bit of the different steps I had in my PhD. So first we started with studying the impact of nursery degradation for one stock, which had a metapopulation structure. So it was a very, we're using a simulation model, which was a spatially structured life cycle model, which was fitted in Bayesian framework. So a very specific and precisely parameterized model, so which requires a lot of work for estimation and before making simulation. And we're quite happy with the results we got with that model, but still a bit frustrated because if you want to do another case study, you have to rebuilding that, and that's a lot of work. So we wanted to step away from this and get a more generic formulation, generic framework to be able to parameterize the model for a lot of species and multiply case study. So I did develop a model like this in my PhD, and I used it to answer a few questions. So do the population response to nursery impact differ along a gradient life histories? Then we applied it to a case study. So we were interested to look how does the coastal degradation impact a community of commercial species sharing the same nursery ground. And finally, I looked through how the population response impact on nursery and spawning ground differ, and how this is related to life history trends. So first, I'm going to explain a bit about the design of a generic simulation model I developed. So we wanted to keep it very simple. So we are representing one population with one nursery ground, one spawning ground, like no spatial heterogeneity, no population structure. And we use very classical equation for adults. I'm not presenting it here because you know them. And I'm detailing a bit more the stock recruitment relationship, so which gives the number of juvenile produced by, let's say, eggs, legs. And we use the Beaverton-Holt stock recruitment relationship because it's explicit compensatory density-dependent mortality, which are happening in the nursery because of the competition among juveniles for food and for space. So this well-known expression is based on this differential equation saying that the number of juvenile is related to a density-independent mortality term, MDI, and a density-dependent mortality term, MDD, and also related to the surface of the nursery area, surface area of the nursery. So if you integrate that over time, you get this nice curve, asymptotic curve that we all know, and you can parameterize this with population parameter. So carrying capacity of the nursery would be the maximum juvenile you are producing in one nursery, and the steepness, which gives you the curve, like the inclination of the curve, is a proportion of the unfished recruitment produced when the stock is reduced to 20% of its initial biomass. And so if you do the math, you can see that the carrying capacity and the steepness are function of those mortality rates and surface parameter that you get in the differential equation in the beginning. I'm putting this because we're going to use that later. So the simulation framework is quite simple. We're using this model. We are aiming to have parameterization of the model accessible from stock assessment, data, and literature. We're using the model. We are designing some habitat scenario that I will mention later, and we're looking at the output. So we're doing long-term simulation and looking at the equilibrium, stock spawning biomass and cash, and trying to building some production curve to find the maximum sustainable yield, and using the indicators of CMSY, FMSY, fishing mortality at MSY, and SSB MSY. So now the core of this chapter was designing the juvenile habitat scenarios. And so first you can think about habitat impact on the surface area of the nursery. And so you might say that it can be decreased, for example, with construction or anything. And so this, if you're modifying the surface, you would affect directly the carrying capacity that I mentioned earlier, because it's directly related to that parameter. You can also think of quality degradation. So the quality is a bit less intuitive or harder to define a component, but it's related to the nursery function and how it's supposed to support juvenile growth and survival. And so our hypothesis is that the juvenile won't avoid the nursery even if it was impacted by the pressure, because they have low swimming capacity and that they really rely on this area. And so that can happen. We can have direct and indirect impact on the juvenile, which would increase the juvenile mortality. So the two terms, density independent and density dependent mortality, which would then generate change in the carrying capacity, but also in the stiffness. And how I'm modeling that, not technically, but operationally, I'm using what we call multipliers. So clear lambda quality for quality scenario and lambda surface for surface scenario, and mitigating value between 0.5 and 1.5 to model degradation or restoration. So some general results about the nursery modification, starting to look at the modification of stock recruitment parameters. So here for a surface scenario, as I mentioned earlier, those scenarios are only modifying the carrying capacity of the nursery, not the steepness. While for quality scenario, you're modifying both, with increasing when restoring and decreasing when degrading. And what's of very interest for us, it's what is the consequence on population dynamics. So we are looking at that for production curve here. So in black, we have the baseline scenario, so baseline production curve. And here for the surface scenario, you can see that modifying the surface of the nursery is just modifying the magnitude and the size of the curve. So you would modify catch at MSY and SSB at MSY in turn. While for quality scenario, you're also modifying the shape of the curve. So you're also modifying the fishing mortality rates, which give you the MSY. So then we wanted to apply this to contrasting life histories. So we look to basis to have consistent origin of data. So we use fish-based and fish life. And to maximize contrast between life histories, we did a PCA on some life history traits I was using in the model. So here's the results, and we're kind of picking species which are highly scattered on this 2D, and for which we have enough data then to parameterize the model. So we choose 3 species because they are highly scattered, and they are also, we're not interested in this precise species. We are more interested in the characteristic life histories, characteristic they are representing, kind of archetype of life histories. So we have anchovy, which is short-lived, high fecundity, and medium stiffness. The the place, which is long-lived, high fecundity, and high steepness, and the cuckoray, which is long-lived, low fecundity, and low steepness. So for the fish species, we're gathering population parameters from fish life, fish base, and a few other sources, and we are setting the same population size for the three population. We are setting the same carrying capacity, which in turn gives the size of the population. And in the simulation, we also add a bit of parameters uncertainty for the parameters extracted from fish life. So we kind of replicate a number of population for each species, and then to have an idea of how the distribution impacts the distribution of the results. I'm not giving a lot of detail on this, but I can give more later if you want. So now we are looking at the multi-species comparison of the results. So for surface scenario, you can see that the steepness is not affected by this scenario. I already mentioned that earlier, but the carrying capacity is, but the results is the same for the three species. So there's a similar impact, that's why you only see one line. And so the consequences at the population scale are going to be the same as well. Well, for quality scenario, you have some interesting differences between species. So you can see for steepness, that the ray is the most sensitive population, while anchovy and plaice are more mixed and less responsive, and then plaice would differentiate a bit for high level of degradation. Looking at the carrying capacity, you can see that it's plaice being the more sensitive and anchovy being the less. And now we want to see what are the consequences on the population dynamics. So here I'm presenting the result for C at MSY, catch at MSY. And you can see that ray is species which is the most impacted, then plaice, then anchovy. And this is because this sensitivity of the CMSY is a bit of a mix between the steepness sensitivity and the carrying capacity sensitivity. So just as a summary of this, for the surface area, we have the main response in the population for the three species, and for quality, we have a gradient of sensitivity, which is mostly driven by the steepness value, initial steepness value. Not only, there is also other life history traits which are at play, but mostly the steepness. So then we wanted to apply this framework to a real life case study. So we are having our case study in the Eastern English Channel, which is on the French coastline, and studying here 4 stocks, sole, whiting, plaice, and sea bass, which are distributed over this area and which are of high fishing interest. They are having common nurseries area. So you can see here intensity of juvenile maps for sole and whiting, but you can see that the maps are not exactly the same, and this is because the juvenile has different bathymetric distribution. So here you have the percentage of fish in the very shallow under 5 meter area. So you have very few whiting and a lot of sea bass. So this is an important feature for later. And those 4 species also have different life histories for sure. So the simulation framework is mostly the same. This time we are picking population parameter in the stock assessment report from ICES. And we want to set carrying capacity, which is realistic compared to the size of the population. So we are doing a small calibration process by minimizing difference between stock assessment report and the model outputs. And the most important thing, we using the same settings for simulating habitat scenarios, but we want to build some realistic values of the scenarios. So we want to get realistic values of land locality and land surface. And to build that, we are for surface using some historical bathymetry map that we have. So we're comparing the availability of shallow area under 5 meters in different estuaries. So from the so estimating the difference between 1870 and 2020. So this is for Seine estuary, and we are doing this for 2 other estuary. And so we have a mean of a loss of those habitats of 38%. Yeah.
Can you say that again? Like, how do you know 1870 what the?
So we have a map of the bathymetry at that time in the estuary. And so we are comparing the quantity of shallow habitats, which represent the nursery for the.
And is it like direct?
Yeah, in those estuary you had a lot of dredging. You can have also like modification upflow, which would modify the hydrodynamic of the system. But it's mostly dredging and construction, dikes and everything. The French estuaries, especially in this area, have been modified a lot. So we have an estimate of the surface loss for this historical period, and there is, for quality, we're using a study that look at the density of juvenile in different estuaries and comparing the density for good, moderate, and bad quality of the water. So mostly in relation with pollutant. And so this study showed that we lose 20% of density of juvenile from a good to a moderate estuary. So we want to build some restoration factor. So we are looking to increase our density 20% of juvenile and 38% of surface as well. And as I mentioned, for surface, we are looking at the very shallow area. So we have to mitigate our maybe weighting our restoration factor according to the species-specific fish distribution in those shallow area. So you remember it was very few fish for whiting and a lot of fish for sea bass. So that's how we get some species-specific restoration factor. And so this is also making the hypothesis that the quality of the water is more degraded in the 5 meter area than on the old nursery. So yeah, that's what the modeling framework looks like. We are imputing our realistic habitat scenario and looking at the results at MSY. And so here I'm showing the catch increase at MSY compared to a baseline scenario for surface quality and combined scenario and for the 4 species. So you can first see that the surface have a larger effect than quality scenario, and that combined scenario has a light synergy. And you can also notice that there is the sensitivity in the response of the population is mostly driven by the concentration on low bathymetry, which make a lot of sense for sure. So sea bass is the most sensitive species then. And so because this results is really impacting, like this hypothesis of which fish are the most impacted really drives the results. We also did like a sensitivity analysis for the quality scenario. So on the left, that was I just presented, that we are considering that the most impacted area are the one within the 5 meter band. But this is a proxy. We cannot, it's hard to say which are the most polluted water and in what extent the pollution is going. So we can also say the pollution is widely distributed over the whole nursery area. So this would be like not using this bathymetric distribution. And so you can see that you are changing a lot your results. You have stronger response for all species because you're impacting all juvenile and not only a fraction of them. And also the sensitivity of the species are changing. So this time, the most driven factor would be the stiffness. So you have whiting and sea bass, which are responding more than flatfish. And while in this one, it was the concentration on low bathymetry, which was driving the responses. So I'm going to walk you through the fourth chapter, in which we wanted to get an extension of this framework to take into account the impact on spawning ground. So I did that. We started this within a collaboration with Ian Brown at DTU Aqua in Denmark. And so this chapter is a bit grounded in the Baltic context, so for which there is numerous, like a lot of hypoxia events, and they are increasing in duration, intensity, and spatial coverage. And so there is a lot of event happening in the deep basin and in the coastal area, which are spawning grounds and or nurseries for plaice, herring, and cod, which are important fishing species there. So we wanted to model that and look at, do population response to impact on nursery and spawning ground differ, and how this is related to the life history trace. So we're using still the same modeling framework, and I'm going to show you a bit more about the scenario because we are expanding our scenario theory. So for the juvenile impact of hypoxia, we are assuming that's the same mechanism than for quality scenario. So juvenile cannot move, they are not avoiding this impacted area, so they have increased mortality, which is impacting both stiffness and carrying capacity, just like before. And for adults, like spawners, which are experiencing hypoxia event in their spawning grounds, you can think of two ways of like behavior mostly. So either they would avoid the area, either they won't avoid this impacted area, and this has different consequences on the population scale. So if they're not avoiding this area, they might have an increase in mortality, which would result in modifying the mortality of the spawner in the model. While if they avoid this area, they might spawn in a less suitable area. So you then might increase the egg mortality, which is going to affect the stiffness value. So, and as I did before, we're using multipliers to model this restoration or degradation scenario, so applied on spawners, eggs, or juveniles. And this is the result for this. So we're looking at the catch increase at MSY compared to the baseline scenario for egg, spawner, and juvenile scenarios. So the first thing you can see is that you have a much larger effect of the spawner scenario and very less of the eggs. And you can also see that the sensitivity of the species is changing within scenario. So for the egg scenario, herring is the most sensitive species, and this is really related to his lowest stiffness value compared to the other fish, to the other population. For spawner scenario, plaice is having his being the most impacted, and this is because it's the longest living species within the three, and so you have all those age class which are mature and having a big weight in the population numbers which are affected. And for juvenile, that's the cod, which is the most sensitive species. So in the end of this study, we have kind of a balanced feeling because we had two hypotheses we couldn't really decide. Either it's the eggs which are impacted or the spawners, and the sensitivity is really opposed. Like in one, the plaice is the most impacted, and the other one, it's the less impacted. So this is kind of I don't know how to say that. This is difficult, like it's so what I want to say is we have two hypotheses, and we also know that probably both hypotheses are happening at the same time within a population. So it's still hard to balance which would be the best or which modeling like hypothesis is the best. So that's kind of a big limit for this study. So I'm going to show you a bit of a summary of the results of this thesis. So we were looking at the modification that habitat impact would have on the population dynamics and population productivity. So for nursery, we saw that impacting the surface is modifying the stock size and the fishery yields at MSY. And if we're modifying the quality of the nursery, we're impacting stock size, fishery yields, and also fishing mortality given the MSY. And this is quite comparable to some results from the literature. For spawning ground, we have those two hypotheses about eggs or adults impact, and both are having impact on stock size, fishery, and harvest rate at MSY. And adults scenarios have a larger impact than egg scenario. And the egg scenario is kind of numerically the same than changing the fecundity of the fish. And it was already observed that this is not having a very high impact on the population dynamics. So we were not very surprised by this. We're also interested in looking at the difference between life histories among those impacts. So for nursery, we emphasize that for surface, it's mostly the concentration on nurseries area which is driving the response. And for quality scenario, that this, but also the steepness and other life history traits, which are driving the population sensitivity. And for spawning ground, we still have those two hypotheses and different driver steepness or the offset of age at maturity and maximum age. As I mentioned earlier, it's a very simple model, so it has a lot of limits and simplifying hypotheses. I'm going to highlight a few of them. It's a deterministic model, so we are missing a lot of the stochastic dynamic that we know it's happening in the real life. We used some parameters variability within our simulation to get a bit of this, but we are still missing some process stochasticity. And we can obviously think about the stock recruitment process, which is known to be very noisy. And also the, how to say that? The intensity of the noise of the stock recruitment relationship is known to be related to some life history traits or life histories, strategies of fish. So that could be an interesting point to look for in the future. We are also ignoring the population spatial structure and the heterogeneity of the space. And that could be very interesting to a very interesting venue to look at with the interaction with scenarios, habitat scenarios, because obviously we're studying habitats. So there is possibility to, so we didn't do that because adding population spatial structure and increasing the complexity and the parameterization complexity a lot, but that's an interesting research feature, and it can be done kind of step by step in scenarios. That's a possibility. And most of all, I would say we're missing some link between the pressure and the impact, like in pressure and the impact of the population. So that's why what we try to model, but in the end, we are missing the data, the population, a long-term data survey, which would be able to compare to the long-term trend that we are simulating. So that's one of the big limits of this, that we cannot really validate the simulation with that. Finally, we were looking to build a transferable model, a generic model who can be applied to a lot of case study. So there is numerous data available for model parameterization. There are some data available for parameterizing scenarios. So we use some of them, but there is, depending on the case study, you can think of a lot of things. And it's also possible to extend the habitat scenario, thinking of other habitat degradation, maybe climate change or biological invasion, which we could think. And the aim of this model was to inform management. Of course, it's not aimed to be embedded in stock assessment process because we are projecting long-term trends, and that's not what stock assessment is looking to, but it's more about fitting spatial planning maybe, and balance between conservation and restoration goals. So that's all for this presentation. I wanted to do a slide about my postdoc at SAFS, but I really missed time about this. So as Christine mentioned, I'm here at the Punt Lab. I'm working with Cole Monahan and Jane, which is remote in Alaska, at the Alaskan Fishing Center of NOAA. And we are going to work about some drivers of productivity for Gulf of Alaska species, and with using state-space stock assessment model. So yeah, that's all.
So much.
Thank
We'll
you.
take a question from the room first to give folks online a moment to type their question into the chat box. But yeah, if we'd like to start, you got to ask a question. So let Andre go first. I think we're gonna hold down. Andre.
I actually like deterministic models because you can write things down. So I have a couple of questions, two linked questions. I'm going to ask two questions in. The first one is related to the quality affecting both MDI and MDD. Is there any evidence to assume that that is the case? But obviously, if you adjust MDI, that's just going to affect steepness. It's not going to affect K. It's just that. So that seems to be a key assumption, is how those the two types of density M's are affected by quality. And then the more sort of esoteric question was, did you ever derive the implied stock-fruit relationship? Because a yield function is no more than yield per recruit times spawning biomass per recruit. You don't need to do a simulation, you just write it down. And so I was really keen, I couldn't do the integration in my head, but I'm sure you can write down the yield, the stock-fruit relationship in terms of lambda serve and lambda quality, in which case you can actually see what's driving the answers. You don't need to run a model, you can just, there's an equation which has those parameters in. I was wondering if you derived that at any stage, because I think that would help to understand what the answer is before you do any fancy computer stuff.
Yeah, so thanks for this question. I'm starting with the second one. So yes, we were at a point aiming at doing this, and we were kind of we were trying to how to say that, to base our analysis on very old Moussaline and Hilborn paper, which were doing this, like very analytical analysis.
Come to my class in fall, we'll do it during class.
Cool. Yeah, and I don't know why we couldn't get we never managed to get it written all well. So that's would be very interesting, because of course we're kind of trying to write some mechanistical relationship. So that's a bit frustrating that you don't get this to the end. So we were I've been analyzing a bit the derivative for stock recruitment parameters, and then we're also looking at the FMSY sensitivity was related to steepness derivative relative to steepness value. So we've been a bit interpreting this in that way, but couldn't get the whole model resolved in that way, which would have been very nice, of course. And the first question about the quality affecting both MDI and MDD. What was like for us? I think there is enough evidence that both are affected, because like it's kind of obvious that both individual mortalities are saying you're increasing pollutants, so that's going to be so if you're doing some in-lab tank, you can see that you can increase some individual mortality, and the density dependent term would be more related to the prey availability, or because the pollutant might also affect the whole community. So this is in this way, but what is so in our opinion, like both terms are impacted, but we're using the same multipliers for both, which is a very big assumption. And so this is related to what I was mentioned in the limits. We really lack data on the field, I would say, that can give us difference about impact on density dependent and density independent data. So we're using the same, and we know that's not the more realistic way to do it, but we didn't have better to do.
Jim.
Yeah, thank you. So yeah, it's super interesting to see this. And I was gonna say, you know, I've been involved with work in the Southeast Center, like for brown shrimp and coastal restoration, you know, where they're interested in similar things. I was just gonna ask about the policy and management kind of system in France. You know, like, was this project initiated because people are interested in informing restoration targets? Or, you know, how is EFH sort of used in the IC system to, you know, like where does this information go?
Yeah, thanks for this question. So I don't have a slide for this, but the PhD was funded by the French Office for Biodiversity, which is like a French entities, which is encompassing, like embedding some forestry things, some MPA, marine protected area as well. And so kind of wildlife monitoring and managing entities, and the stock assessment is done, and the management is done at the European level. So there is like one of the biggest problem, I would say, that there is it's different entities which are not really talking together, I guess. So like in the stock assessment, this is not taken into account at all, either the habitat related or mostly like environmental. It's still, I guess it's beginning, but it's still hard to get there. And yeah, so the OFB funded the PhD because they were it's also it was a tool. It was also a tool for them to show what would be the difference for different species, what would be the outcome for different species of the restoration, for example. And it's also making the link between individual and population scale is making more strength in the plaidoyer to support some restoration or not degradation, because you can relate this to kilograms of fish. So that's kind of, even if we are not always showing so in the case study, we could relate this to actual cash or things. So that was interesting for them. So, yeah.
Any other questions?
Um, so you had a certain range of habitat changes that you considered degrading and improving.
Yes, we.
And how did you pick those values or proportions?
So in the more theoretical studies, this like study I did, this is really like increasing or decreasing 50%. Like we are just making a range of plus 30%, plus 50%, less 50% to see how this is impacting population. And so for the case study, we actually link those restoration factor to data we had on the field to know, like, so I was showing that's a long.
So the changes were symmetrical, right? So they were plus or minus 50% with some reference values, something like that. You had some graphs where you showed the
Yeah, I think you're speaking of those, which are kind of, yeah, so you have, so it's not totally symmetrical. Yeah, so yes, the 25%.
Yeah, so my question was, Andre, I'm sure will correct me if I'm correct. My understanding is if we look at abundance of fish populations, they generally follow a logarithmic distribution. And that means, if, to offset a decline of 50%, you don't have an increase of 50%, that won't get you back to the normal. You need to increase of 100%, doubling. So that would suggest that you might want to have this range of scenarios asymmetrical. So on the increasing side, you might want to have it, say if it's 50% below for the decrease, you might double it for the increase side. Would that make any sense?
Uh,
Well, it's a deterministic model, so there's no stochasticity in here anymore.
Yes, so I think I understand your question, and it's mostly related to the
Okay, here's the point you could have made, and that is the bottom right corner of that figure, which shows the variance changing as a function of how the parameters are changing. And the reason I think for that is that steepness is a very nonlinear parameter in the yield function. So you could, if you're at 0.9, you can't get much more than 0.9 because you can't get bigger than 0.1. They get flat, right? But obviously you could reduce steepness down to 0.2. So if you've got stock that, in the case of the group, you've got low steepness, so you're on just the really steep part of yield function.
Yeah, that's why we narrow the range of scenario actually, because when adding the uncertainty and like the degradation stuff, we were falling under 0.2, which is unrealistic, and same for restoration with too high scenario, we're above 1, which makes no sense as well. So
shouldn't be able to get above 1. Mathematically.
but doing like just multiplying, using the multipliers, you could get above, which makes no sense. So we didn't do the simulation.
It's algebra. German.
I don't see any questions online. They have about 30 seconds to type that in. Otherwise, I want to thank you. Oh, wait, Enrico.
Very general about the can. There's a lot of interest to try to understand what happened to the learning facility. So, in your discussion, this study, you see how the dynamic of what is happening in reality is actually matching your prediction, and
the habitat degradation is matching the playing there.
I would say yes and no, because we are like modeling a very theoretical population of herring. So I guess it's so. I think it's missing some realistic feature to really match the trends that we would have in the Baltic Sea. And that's also, I guess, the problem that a lot of factors are at play in the herring system in the Baltic. So it would be hard to say that this is only related, like this is the habitat having impact there or not. But that's an interesting. I see your point, and so we don't have that much of a link to the real world stock, I would say, in this study.
The question is, what was the impact of mulberries?
Of
Mulberries in that particular area.
Melbourne.
You know a mulberry, 1944?
That's my date of
When
birth.
Operation Overlord, the British built those a huge amount of habitat, they only destroyed the habitat of the northern part of France where the landings occurred. Built you look at the mulberry.
I typed that comment into the every time any questions to people online. I'm gonna go on with some fun historical knowledge. And with that, well, let's thank Juliette one more time for presenting. And for this next week, Enrico will be sharing some of his PhD work in this same place, same time. Everyone have a nice weekend. Thank you for coming. Thank you.